We shall record. Okay, so to finish everything off, guys, we talked a lot last time about the evidence that is out there on the parameters, if you will, of human resistance training. So the effects of intensity and the number of sets, the number of reps, the contraction type, things of that nature on kind of how we observe hypertrophy, okay? So we're going to finish off everything with all of this by talking in a little bit more detail about the actual underlying cellular and molecular mechanisms of this. So I know that we talked about a couple of classes ago, we talked about the idea that there's these signaling pathways and we need to activate stuff in the nucleus, and then that's going to lead to the formation of certain messenger RNA, which are then going to get translated at the ribosomes into proteins, okay? So what I'm gonna show you guys now is some evidence on what we can see as acute changes in actual measured protein synthesis rates. So if we do some things, we're going to measure kind of whole body protein synthesis, and then we'll kind of move from there down to what we think some of the more well-known cellular kind of signals and players and all of this are going to be. Okay, so as we mentioned, right, there's this very complex and integrative signals that are going to control myofibrillar protein synthesis. It's not just one signal, okay? It's not just one thing. But so we're going to have some problems when we try to study this, though, because issues are going to arise when looking at some sort of an acute response. So we send Alex to the weight room today, we have him do one thing, and then we see what changes, like what cellular pathways may be activated immediately afterwards or within a couple of hours afterwards, or we see what's happening at his whole body fractional protein synthesis rates or something after one bout. We can measure all of those things, okay? But we see a disconnect at times between kind of the magnitude of what the acute response and the acute signal is, and then what we're going to see if we do training for a much, much longer period of time, okay? What we do know, okay, is that a single bout of training tends to lead to a positive net protein balance. We're going to get to a place where synthesis is going to tend to exceed degradation. And some of that, it's this weird process. Some of it may be that synthesis stays the same and we blunt degradation afterwards. In some instances, they observe that both synthesis and degradation go up, but synthesis goes up to a greater extent than the increase in degradation. And so the net out of all of that is that we're going to be in a more positive protein balance. So the idea then is, well, something has to be driving or causing this. There's something about what you did today that activates certain pathways that are then going to cause this increase in everything, okay? So here's some data that were published in 2005, and these again are out of Bob Wolf's lab at the UT Med Center in Galveston, Texas, where the vast majority of kind of the protein synthesis rate stuff is going to come from. They do really, really good work there in Galveston. But this is myofibrillar fractional protein synthesis, and we're not going to talk about what fractional synthesis rates are versus some other things, but just note on all of this that here's their resting values. They had them perform a single bout of resistance training, and what they noticed out of all of this was that there was this increase, right, kind of progressively over the first 24 hours, and then it began to taper back off again. But even at 72 hours post, we still had elevated protein synthesis rates compared to rest, okay? So there is some evidence that resistance training leads to maybe up to a 3-day increase in protein synthesis rates. Now, some of this is going to be partially controlled by if we give you a lot of protein and stuff in the aftermath of all of this. Plus, I can just give you protein. We could just feed Megan a bunch of protein, okay? If you do all of that, then you will also get an increase in protein synthesis rate, but it only tends to last for maybe 24 hours. There seems to be some additional increases that we're going to see based upon all of this, okay? So Weirdly, in a lot of instances, after resistance training, you get an increase in synthesis, but you also get an increase in degradation. And some of that is that we have damaged some of the muscle, and so there's degradation as that gets broken down. Other parts of it may be due from the fact that in tissue or muscles where we weren't doing resistance training, that there's going to be an increase in the breakdown of proteins there so that we can have amino acids to be able to incorporate into the muscle that we just stimulated via resistance training. Okay, and so this is why we think it's really, really important that you need to make sure that you are eating a sufficient amount of amino acids in the kind of days after doing resistance training, or while you're doing it, that you need to step all of that up. That seems to potentially blunt a little bit of the degradation response and have some synergistic effects with all of this. Does that make sense, guys? Okay, so all of that's going to go up. Here are some additional data that we're going to see, and this is a weird thing where you're going to see that afterwards that synthesis rate goes up, breakdown rates are going to go up. The net balance, though, is going to be a little better off than it is here. Please don't ask me to try to explain everything in this graph. It makes my head hurt to watch all of this because somebody always points out, Dr. Black, how can your net protein balance be like negative? And that's still a good thing for hypertrophy. And I'm like, well, but it's less negative than it was at rest. And so that is technically a gain. These findings have not, in this particular manner, been exactly replicated like all of this kind of going forward. But this is from the same paper. This is the fractional synthesis rate increasing. So don't worry about the units. The units don't really matter that much, but we do see kind of this increase, especially kind of 3 hours post. Okay. All right. So what do we think are some of the things that are going on that are going to drive this increase in synthesis rate, or potentially kind of blunt or maybe attenuate this increase in degradation? So there is a school of thought, and we do have some evidence to support all of this, that there are these anabolic hormones that are going to be released during exercise, but they're also very often going to be circulating. You're just going to have some of these things that are going to be there under normal circumstances. And those typical anabolic hormones are testosterone, growth hormone, something called IGF-1 or MGF. And these are actually separate hormones. They're different variants of each. IGF stands for insulin-like growth factor 1. Okay. MGF stands for mechano growth factor. And so MGF is a particular variant of IGF-1 that is produced only by muscles. And we think that MGF is particularly important for activating satellite cells. And so it's particularly important in the hypertrophic response that we see in response to resistance training. Okay, so I don't want to make too big of a deal because I feel like that our field has evolved to some extent. When you exercise, testosterone goes up. When you exercise, growth hormone goes up. When you exercise, IGF-1 and MGF tend to go up. The problem is that these things all tend to go up in response to aerobic training, but also resistance training. And so this idea that I do resistance training, therefore I'm going to release more testosterone and more growth hormone, and those increases in the hormones are therefore going to drive some increase in protein synthesis. Well, there may be a part of that that's true. It's not nearly maybe as kind of the sole mechanism as what we used to think. Okay, and I'll explain a little more about that in the next few slides. Okay, so much of what we know about these hormones and their effects on protein synthesis are going to come from people that generally don't have enough of this hormone, and then we give them some hormone back, or people that are just completely deficient because we don't have it's in some ways very unethical for us to give people these hormones. Okay, so what we know is if we take men that are what we call hypogonadal men, so these are men that do not produce enough testosterone. Okay, if we give them a physiological dose, if we give them testosterone to bring them from being low back up to a normal kind of age-associated level of testosterone, we tend to see about a 15% increase in muscle mass and about a 50% increase in protein synthesis rates in those men. Okay? If we supplement, this is without resistance training, this is with nothing else. If I just give guys that don't make enough testosterone additional testosterone, voila, their muscles magically get bigger. This has been very, very, very limited in study, but if I supplement and you go down, so if I take guys that make enough testosterone and I give them extra, they may see with doing nothing, maybe somewhere between a 5 to a 20% increase in mass. Okay? If you take extra testosterone, you're going to get swole. Period. Full stop. End of discussion. Okay? If you take extra testosterone and you combine that with resistance training, you're going to get really swole. Okay? We may not even understand fully like what the limits on all of that are going to be. And so we know a few things that we know that testosterone is important. Okay? But the more kind of meaningful piece of all of this may be that if we give it to you extra, we know it drives synthesis, but there may be something to the fact, the kind of very simple fact that as long as you have enough, kind of some normal amount of it floating around, then you're going to get some kind of an appropriate response following a bout of exercise. There's not a lot of good evidence that the acute change after a bout of exercise is in some way, shape, or form related to the amount of hypertrophy that we're going to see. It's not like the guys that have a bigger testosterone response all seem to show a larger hypertrophy response or something like that. Okay, so it's a kind of a tricky thing. So similarly with growth hormone, we know that if we supplement growth hormone in individuals, we tend to see increases in muscle mass, increases in protein synthesis, and decreases in breakdown. If we supplement growth hormone and we do resistance training, we don't see a lot of additional gains. Okay, and one of the problems, especially with growth hormone, is that it's difficult for us to tease out the independent effects of growth hormone from any kind of effects on IGF-1, because when your growth hormone levels go up, your IGF-1 levels go up. Also, when your testosterone levels go up, your growth hormone levels go up, and your IGF-1 levels go up. So there's this difficult pattern of being able to sort of tease out the individual effects of some of these kinds of things. Okay? So IGF-1 is made in the liver and in the muscle. So the liver portion is just called IGF-1. You have the exact same form of it that also comes out of muscles, but then you have this IGF-1 E(c) variant that we call mechanogrowth factor that's only going to be produced in the muscle. Okay, and especially MGF, we know is going to be released by skeletal muscle in response to both stretch and overload. And MGF, if you inject it directly into a recently exercised skeletal muscle, is going to activate satellite cells, and it appears to increase protein synthesis to an even greater extent than kind of the IGF-1, the regular IGF-1 from the liver, and the regular IGF-1 from skeletal muscle. Okay, so this one very often we think tends to act in an autocrine and a paracrine fashion, that you work out and you release all of these things. But again, a lot of what we know about these and its role are from people that don't make enough of it. Okay, and so it seems to be one of those things that is maybe helpful, but not kind of the whole deal. If weirdly enough, if you want to supplement MGF, you can't supplement it, like you can't just inject yourself in your butt, or you can't take an IV of it and get this systemic infusion of MGF and get any kind of actual gains from it. What you need to do is you need to work out, and you need to inject it directly into the muscle that's just recently worked out. Contractions tend to upregulate receptors for this in that particular muscle, and it also seems to kind of make, there's something about that autocrine paracrine action of the MGF that seems to be really, really important, even though we don't fully understand all of that. Okay, so what's going to be some of the stimuli to release these mechanical hormones? We know that muscle loading and stretching, as well as the total muscle mass use, are all going to play a role in kind of driving this release in these things. How to stretch and loading stimulate release, we don't fully understand, but there definitely seems to be something about mechanical stress, maybe mediated by calcium release or something that's placed along the outer membrane of the muscle, and that are going to help us release hormones from that cell and drive the upregulation of receptors for that hormone in those particular cells. Okay, so we know that this is important, but it's not maybe the end all be all that we once thought it was. Okay, so Alex asked the other day about kind of moderate weight training and high weight training and those kinds of things. And I think I told you guys that there's this big misconception based upon some data that it kind of dominates the NSCA's recommendations for things. Okay, and so let me see. Keldon's still here. Yep, Keldon, Ryan, Alex, I know y'all are some of my resistance trainers and those things. If I told you guys we need to do hypertrophy training, what's the first thing that comes to mind for you guys? What's that going to look like if I want to do hypertrophy training versus like strength training?
I mean, traditionally, especially if you look, I mean, speaking of recommendations, traditionally it's like the typical 8 to 12 reps, 2 to 3 sets. I can't remember the percentages. I know that ACSM and NSCA are a little bit different in terms of that. It was like 60 to 80%, somewhere in that range probably. And then I guess like the traditional, after that's like power is like the 3 to 5 set range, and then strength or maximal strength is like 1 to 3 reps above 90%.
Okay, Alex is on to something. Keldon, Ryan?
I always say like more of like a low volume, higher rep. I think that's what I think of whenever I go to hypertrophy.
When you say lower volume, high rep, can you be more specific, Ryan? Like, can you give us an example?
I could probably say like for me, if I was doing squats, I wouldn't do obviously I would do a weight that's like kind of hard, but still kind of pushes, but it's lower volume than like my 1 rep max. And I'd go for like, as Alex said, like 8 to 10 reps.
Okay. Any other thing? Do I have anybody that's an NSCA, like a certified a CSCS or anything about when they talk about hypertrophy, they also talk about manipulating a variable we haven't talked about yet?
Time under tension.
Time under tension, Keldon?
Yeah.
Okay. I'm thinking of something else too, though.
And then there's also like reps to failure.
Reps to failure, okay.
Are you thinking of rest in between sets?
Yes, Megan, I'm thinking about rest in between sets.
Just so you know, I have SCS, so you can include me with the other group who's in the resistance training.
I include everybody in there. Sometimes I want to see what other people know, Megan. So you're a CSCS. Okay. So what are the rest period recommendations for hypertrophy training versus, say, strength training?
I think hypertrophy, you're looking at like 60 to 90 seconds, where strength training 2 to even upwards of 5 minutes, depending.
Mm-hmm. You ever tried to train that way? Anybody ever tried to wait 60 seconds between sets when you're doing things? I mean, you've never done that?
Well, I usually do more strength geared training, so I'm usually at like 2 or 3 minutes.
Okay. It sucks. I'll just say that. Like you're already fatigued when you start your second and your third set and those kinds of things. And so there's this idea, and it has been kind of widely published or widely publicized, I should say, that if you want to do hypertrophy training, that it needs to be this kind of intermediate number of repetitions, like this 8 to 12, 2 or 3 or 4 sets, and this kind of intermediate weight, intermediate rep numbers. And they promote that you want to have these relatively shorter rest periods in between sets. All of this is based in large part on the data that you see right here in front of you. So when I say this is the origin of all of our issues with some of the hormonal response and its role in hypertrophy, a lot of it is based upon this. Now, this is a paper by Bill Kramer. I think Bill's the chair at Ohio State now. He moves around so much it's hard to know. He's been the chair at Ball State, the chair at Penn State and things. He's been in Connecticut. He's been in a lot of places. He's really, really good scientist. But this particular paper they were looking at the acute growth hormone response after different kind of loading and rest schemes. Okay. And so this piece here, where you see this giant increase, this is growth hormone that's up. Okay. And so this is looking at the amount where they're doing like a set of 3 sets of 10 where they're controlling their load, 5 sets of 3 where they're doing some things, and then 5 sets of 1, which is kind of their control. This one is higher reps. So it's 10 reps, 1 minute of rest versus higher force, 5 reps, 1 minute of rest. Okay. And then 10 reps and 3 minutes of rest. The big thing here is that, look, I'm doing higher reps, a short rest period, and I get this giant, in comparison, increase in growth hormone. And so they took from all of this right here is the thing upon which the hypertrophy training in every NSCA kind of recommendation is based. It's this. It's this idea that having that shortened rest period in these things gets this much bigger growth hormone response, and then the presumption that that bigger growth hormone response will in fact result in more hypertrophy. We now have some data that the acute hormone response does not, in fact, always equate to more kinds of gains in those things. Anyway, so Bill was one of the founders of the NSCA, along with Andy Fry and my former PhD mentor in those things. So, a very bright guy, and he would even tell you now, I've seen him do a presentation at various conferences, he would tell you that this was maybe overinterpreted at the time on some of those things. Okay. All right. So here we're going to have acute changes in fractional synthesis rate and hypertrophy following resistance training. And I'm going to show you guys a little bit of all of this in an effort to kind of think about, is there going to be some kind of effects of hormones or something, and whether or not it's the acute change or whether it's not, it's just actually having some hormones. So here are some data in young people and old people. It's young men and old men. These are old men who do not produce nearly as much testosterone as the young men. And here's their acute change in fractional synthesis rate. So you see this big increase in the young men, almost no increase in the old men. But then if we look at all of this and we look at changes in type 2 fiber hypertrophy after training, they're not getting the changes in synthesis rate, but yet they're getting a relatively equal percentage-wise increase in their change. And so there's some questions about not only the hormonal response and the hormonal response that may be driving some of this kind of observed changes in fractional synthesis rate, but the kind of overall importance of what we see after one bout and how that relates to kind of longer term effects. Okay, I don't have a good answer for this. I just want to present it to you guys as something that is out there that people talk about and kind of have questions about. Okay, so I told you all that there's some data that maybe it doesn't matter, but then here are some data that says that potentially it does matter. And so these are from Marcus Bauman's lab at the University of Alabama, Birmingham. And so you guys will remember that study that I showed you some data from the other day. But here we have, this is the messenger RNA that are being spewed out by the nucleus from inside the individual muscle fibers. And it's mechano growth factor messenger RNA, and as well as the kind of normal regular IGF-1 messenger RNA. These are all coming from skeletal muscle. Okay. Now remember that messenger RNA are not necessarily indicative of actual protein that is being made, but it's a pretty reasonable approximation of this. So if we have messenger RNA, we assume we're making the MGF and the IGF-1 proteins, and then we're going to base those assumptions. If these go up, then maybe those are also driving an increase in synthesis in the kind of surrounding muscle fibers. So they trained a bunch of people, and they did something called a cluster analysis. They took all of their people and they put them into 3 groups, what they called non-responders, what they called moderate responders, and what they called their extreme or their very, very big responders. And so here's when they're measuring this, here's the mean values of their change in myofibrillar cross-sectional area. So you've got your folks that basically didn't get bigger are folks that got this moderate amount of an increase, and are folks that got this giant, very, very big increase in things. And when we look at all of them, this is the sort of acute change in MGF messenger RNA after a single biovariant resistance training protocol. And you will note that in the non-responders, they don't get much of a change. It's up a little bit, but not statistically significant. And the moderate and the extremes are both going to be up, although they're not really different. Then we look at how much of this is still hanging around after 16 weeks of training. And what we're going to find then is that these two are going to be up in comparison to their baselines. But again, the non-responders are still not. They must be cheating with this must be standard errors, not deviations, that this looks small and it's actually not different from here, okay? In IGF-1, there's no acute change differences, but there are going to be some differences where they're all up from baseline here, and that these two are going to be a little bit different than that, okay? So these are some data that maybe the acute response, especially in MGF and IGF-1, are in fact going to be somewhat related to. They're going to be higher in our clusters of people where things are going to get bigger. Okay. I don't have. I have the paper, which let me do that. Okay. And then, so can you guys see this? That I've switched this over. That I've got a paper up now. So here's a paper from 2016 from Stuart Phillips's lab at McMaster, where they found that neither the load that you lifted, so they did high weight and low weight, and they had both of them work out until task failure. And then they also measured the systemic hormone response to certain things. And they found that neither load nor hormones determined the amount of hypertrophy or strength gains that people got. This has since been repeated in a couple of other instances and a couple of other different kinds of experiments. Some people don't love the model that they use to kind of how they train people to kind of get at all of this, but there is some evidence right on sort of both sides of all of this. That's kind of what I want to highlight for you guys. Okay, so how do fibers get bigger? This is going to bring up this idea of hyperplasia. Hyperplasia is the concept that you're going to take one fiber and it's going to get bigger, and then it's going to split into two fibers, and each of those two fibers are going to get bigger. Then they're going to split into four fibers, and then they're going to get bigger. So it's this idea of fiber splitting. Okay, it is basically impossible for us to quantify this in people. It is observed in some animal models, but the best that we can guess is that this does not happen in people. Okay, it just doesn't happen. It may happen if you take steroids, but what we really tend to see in the animal models is that we're going to get it when we do these chronic stretch models of things. Okay, remember that I told you guys that when you undertake this chronic stretch model, you're going to get this giant amount of hypertrophy, but it is in many ways not at all like what we do in each other. So it may be something that is primarily driven by chronic stretch, but we don't really ever do chronic stretch in humans, so we don't know for sure kind of what's going on there. But the best evidence that we have is that it's not actually, it probably doesn't happen in people. Okay, so if that is not how they get bigger, what does actually happen? Well, we are going to tend to get an increase in the size of whatever fibers you do have. And it's going to occur in these two particular phases, okay? During phase 1 hypertrophy, we're going to increase cross-sectional area and the volume a little bit. And then that's going to expand, okay? And then once we get out here to phase 2, not only are we going to, as we get bigger, not only are we going to continue to get an increase in cross-sectional area and volume, but we're also going to begin to add myonuclei. And so there's this kind of two-pronged or two-phase ability. And is there some thought that maybe people that are genetic responders are able to multiply their myonuclei more than other people, or they're able to activate their satellite cells better than some other people? And the answer to that is absolutely yes. We don't have a huge amount of evidence because it's hard for us to kind of really dig in and quantify, because I can't cut Pearson's muscle out and look at it now and reattach it, and then have him train and then cut it out again and grind it up and try to figure out how many myonuclei he has in certain places. And so it's a tricky thing. Another thing that happens, although we're pretty certain it happens when we do resistance training, is there are some fiber type specific changes that are going to occur. Okay, we definitely, and I showed you all data on this on Tuesday, I believe, we definitely see fiber type conversion, where we're going to turn 2X fibers into 2A fibers with resistance training, just like what we would see if we do aerobic training. Basically, any kind of loading, any kind of activity turns the X's into A's. I think we also talked way back about the idea that there are these hybrid fibers that exist in a continuum between the 2A and 2X fibers, and that what we do know is that when you do resistance training, one of the other things that happens is those hybrid fibers all tend to disappear, and they also are going to tend to get turned into 2As. And in some instances, like in these particular data, from the Trappy lab at Ball State, which is one of the, they do some single fiber stuff. It's really kind of really cool, really interesting stuff. It's mostly these hybrid fibers, though, the ones that are getting converted to 2As. And you're not seeing a lot of, in their hands, they don't see a lot of changes in the 2Xs in response to all of this. So happy to share a bunch of this stuff. The Trappy lab at Ball State, it's two brothers, so their last names, both of them, their last names Trappy. It's Scott and Todd. But they work with NASA and a bunch of things, and so they have biopsy data, and they have an ability to take a biopsy and tease out individual muscle fibers, and then they take those muscle fibers and they string them up between these two little posts, and they can measure the strength of the individual fibers and the characteristics of all the individual fibers. And they have that on astronauts, they have it on bears that have been hibernating. They use hibernating bears as one of their models of kind of sedentary behavior and disuse to kind of mimic what might happen with spaceflight and those kinds of things. And they go to Russia all the time, and like they'll get biopsies from the astronauts like right when they land, kind of back down and things. And some really great science that they do. And Scott especially, they're both really nice guys. Scott especially talks at ACSM all the time, and is a really kind of compelling speaker. If you guys ever get a chance to kind of see these things. Okay. So a couple of other things to keep in mind here, okay? Remember we talked about satellite cells way back as well. So we know that satellite cells are these myogenic stem cells, okay? What we see in cell culture a lot of times are satellite cells that we're going to culture. We know that they are important for regeneration and for growth. And part of the way that we know all of this is the fact that if we undertake certain things in animal models, we can basically irradiate the satellite cells, and we can knock out their ability to kind of divide and function and integrate themselves, okay? And so what you can see here, here's some very old data. That sounds bad for me to say, but this data is probably, older than all of you guys here. And what you will note is that these are animal data. They have an untreated control group. This is the extensor digitorum longus muscle in a mouse. And they're comparing it to the contralateral limb control. And they did synergist ablation. So we cut, this is basically the EDL. It's like the plantar flexors in them. So they cut one of the muscles, and the one that is intact gets giant, okay? They then take a group and they irradiate them, and they kill off their satellite cells. They do no training. They kill off their satellite cells, and that muscle gets smaller. They also then take a different group, and they irradiate them to kill off the satellite cells, and then they do the ablation, and they cannot increase in muscle size following the irradiation, even though they've got that kind of chronic stretch from the ablation that's going to be applied. It's going to be there, okay? And so we know from all of this that the satellite cells play some level of a role in the midst of all of this. And if you don't have them and they don't work correctly, then you're not going to show a hypertrophy response in the manner that we would think, okay? So here are kind of some similar data out of Greg Adams' lab at UC Irvine. They were looking at the MGF response, okay? And they think that it's this MGF response in these days that are going to be afterwards here, all right? That are going to be the thing that's going to jack up and activate the satellite cells. That's going to be the big response thing. So this change here is what's driving this big increase in satellite cell activation that's driving the hypertrophy response. Okay. So we'll finish off talking a little bit about some of these molecular signaling pathways. We talked previously about mTOR. mTOR stands for the mammalian target of rapamycin. We name things in ridiculous manners, but that's one of the fun things, that if you discover some new molecular signaling molecule, you can name it whatever you want. So, right, so like Megan could name it after her cat, or her dog or something if she discovers it. We can give them all like fun kinds of things. Like there's this thing that's called RAG, and then there's a thing that regulates RAG, and it's called Regulator. There's a deal that activates on mTOR. It's called Raptor. I think it's always fun to name things in these fun ways. But so if you look at growth hormone and you look at sort of IGF-1 and insulin bind to a relatively similar receptor, you look at some of the downstream products of insulin and the IGF-1 response is that mTOR gets activated. And so it takes some of this ERS1 and PI3 kinase and these kinds of things, and then you're going to get the signaling cascade that's going to eventually come down here and activate P70 S6 kinase that's going to drive the protein synthesis. And so we know in some ways that glycogen depletion activates AMPK, which may blunt the response of mTOR, or it's going to activate this thing that blunts mTOR response, which may be why when you run a bunch, even though you get IGF-1 release during aerobic exercise, this activation of AMPK from glycogen depletion may blunt the response at mTOR, which is why you don't get jacked from doing cross-country running. But then there may be some mechanosensitive thing that plays some kind of a role with these other ones in actually it inhibits the thing that inhibits mTOR, and so then therefore mTOR can get activated, and we're going to get hypertrophy. Okay, so one of the things that we know is that there are a bunch of these kind of downstream activators, and so there's a thing called AKT, which stands for protein kinase B. Okay, which is this molecule right here that also seems to interact with mTOR. And so we know that these are downstream targets of growth hormone, AKT, mTOR, and then the S6 kinase, the P70 S6 kinase. And what they're doing basically is they have a mouse model that it overexpresses AKT. And so it overexpresses this protein kinase B. And you can look at its EDL, and you can look at its soleus muscles, okay? And you treat it with this this TAM molecule, which turns on the overexpression. And so if you just take something and you turn on AKT overexpression, so you're not giving it any exercise, you're not giving it any extra food, but you turn on the AKT overexpression, so you turn on this piece of the pathway, and voila, their muscles get a lot bigger, okay? So are we going to come up with a way to kind of increase Ryan's AKT expression by giving her something? Probably at some point we will, but we know some things from all of that. This is one of the first papers where they looked at we can block mTOR activity, okay? And so this was again in an animal model, but they used the antibiotic rapamycin. So if I apply rapamycin to the cells, or I inject rapamycin into these animals, then the rapamycin stops the activity of mTOR. It's the mammalian target of rapamycin. So that rapamycin binds to mTOR and shuts it off. And so what they did was here we have 7 days or 14 days of compensatory hypertrophy in these animals. So this is a synergist ablation model of doing things. And they get just the compensatory hypertrophy. This is going to be their change in muscle weight. So it goes up almost 25% or so. And then you give them the hypertrophy response, so the synergist ablation, but then you give them rapamycin, so it blunts mTOR, and you get much, much less. At 14 days, again, it's still going to be much, much less. Here we're looking at the actual, this is sort of the muscle weight. This is the individual fiber cross-sectional area. and we see relatively the same kind of thing that works in both fast and slow fibers. This was published in Nature, and it came from a woman named Sue Bodine, who's kind of a big deal in the muscle physiology world in the midst of all of this. Okay? So from this, we know that we think mTOR plays a big and important role. So here we're going to have, we have this idea, then when you want to do these things, that if we can, we have these wild type mouse and these rapamycin resistant mice, and what you find basically is they've genetically modified these animals so that their mTOR does not respond to rapamycin. And so you can basically get this idea where I give them compensatory overload and I give them rapamycin and it doesn't blunt anything. Okay. And so that makes us know that we can kind of do the experiment going in both directions. So here's the fiber cross-sectional area responses, and what you really want to care about in the midst of these things is that this one, which is no rapamycin, and the overload response, you get the biggest response. Here they're getting overload plus rapamycin, and it's blunted in comparison here. And then here's the one where it's not nearly as blunted because it's resistant to the rapamycin. But the cool thing about this particular experiment as well, these were done out of Troy Hornberger's lab at the University of Wisconsin, is that they were also showing that mTOR leads to the activation of ribosomes. Not only does it activate things in the nucleus, it activates S6 kinase, but it also then has some interactions with ribosomes as well. So it's going to make the messenger RNA go up, and then it's also going to increase mitochondrial or ribosomal content in the muscles. And so you need to turn on the gene, but then you also need to turn on the thing that's going to translate the gene into protein, and mTOR seems to do both. Okay, so this now raises the question: does exercise stimulate mTOR? We've seen that some of this compensatory hypertrophy models are going to show us that it does. We know that like growth hormone and testosterone and insulin and IGF-1 activate mTOR, but we also think there is some sort of a mechanoreceptor. There's something that is some sort of protein or channel or something that is likely embedded in the membrane of skeletal muscle that is also going to be sensitive to that loading and that stretching that fiber is undertaking, that is going to also probably independently activate mTOR. We don't fully understand what it is yet, but that's going to get us to there. Okay, so lots and lots and lots and lots and lots of data and interesting things out there. But I just wanted to give you guys just a little taste, a little flavor of kind of what we think is going on. Cellularly, a lot of this stuff is integrated through mTOR. So if there's mTOR, there's Raptor, one of my favorite names. There's RAG, there's Regulator. It's going to help us with these lysosome things that are going to kill off certain things. All right, so we'll finish then with the opposite of all of this. Okay, we'll finish it with something that I call an anti-growth factor. It's called myostatin. Okay, called myostatin. This is a Belgian blue bull. This bull has never lifted a weight in his life, and yet he appears super jacked. The reason he appears super jacked is that he lacks a protein called myostatin. Okay, this baby here has quads and things. This baby, for the most part, lacks myostatin. Okay, myostatin is a naturally occurring protein, and myostatin partially blocks the function of satellite cells. It functions to protect and keep muscles from getting too big and too strong. And so we know if we can blunt or block the effect of myostatin in your body, we can get greater satellite cell activation and we can get greater muscle growth. Okay, we know that myostatin levels are different among everyone in class. There's a pretty good chance that those of you that are bigger responders to resistance training have less myostatin than people that are smaller responders to resistance training. And so like this baby, both of its parents were Olympians. They think neither of the parents had myostatin and those things. And so every once in a while, but there's a myostatin deficient bull and a dog and a mouse and a cat You can Google all of them if you want to, but they all kind of look like this. The poor myostatin dogs just look like it's just huge, ripped up dudes. Okay, so that's kind of the anti-growth factor. So hopefully from all of this, you can see that hypertrophy is complex. There's a lot of signals, a lot of cellular things and signaling pathways that that need to get activated or need to get blunted in an effort for us to kind of increase muscle size and to get hypertrophy. And then there's a lot of other genetic things like how many satellite cells you have, how many myonuclei you have, how much myostatin you have, as well as maybe your kind of normal circulating levels of certain hormones, all of which may work synergistically together to kind of get you to whatever your normal muscle size is going to be. And then we hope with exercise that we can tip the balance one way or the other with that. Okay. All right. That is all from the lecturing portion. So let's talk about critical thinking. Or I guess I should just ask, do y'all have questions about that?
So do you think they'll try to come up with a
I'm sorry, Megan. Can you say that again? You were breaking up on me. Uh-oh.
Do you think they tried to for bodybuilders? I feel like they would like that.
I am certain that there probably exists a myostatin inhibitor that we may or may not be fully aware of that bodybuilders would like to take. Yes. So, and if they don't have one, then I assure you that somebody somewhere is working on one. That's what everybody wants, right? Jacob needs that myostatin inhibitor so he can get real swole. Other questions? I'm having all kinds of problems, guys, with the computer wanting to freeze up on me. So I'm shutting a couple of things down. This. Okay. All right. So let's talk about the critical thinking then. Okay. Alex, read us the first critical thinking question. You're on mute, buddy.
Okay, perfect. If I'm trying to pull up something, we can see too. Okay. All right. So based on everything from this lecture, what would be an ideal or optimal exercise program to increase muscle size and strength?
Mm-hmm.
Defend your answer. So I think this is a really broad question.
Yes,
I think
it
there are
was.
a lot of
room.
different answers to it. I tried to relate this as much as I could to, I guess, some of the things we talked about during lecture. I think there's no optimal approach. I think it's very hard for someone or to have a optimal training program just based on day-to-day changes. But that's kind of the first big thing that I started talking about was it has to be individualized and specific to someone's goal. And then some of the things we talked about is it's like progressive overload. So depending on their goal, which in this case was muscle size and strength. So I just talked about a little bit about using hypertrophy ranges, but then also training maximal strength, and also for the benefit of hypertrophic responses to training for maximal strength. And then, I mean, I talked about a lot of like smaller stuff like stimulus recovery, some of the adaptations we kind of should expect. So I can talk a little bit about everything just because it was such a broad topic. But I think the biggest takeaway for me out of the lecture was specificity, and then looking at individual individual goals and progressively overloading towards those goals.
Okay. Jacob, what about you?
Yeah, I thought this was a very broad question too, especially because I don't do much training. But I pretty much said like I would expect probably you'd want to do enough weight that it would be enough to do progressive overload, such as like, I don't know, 70-80% of your 1 rep max. I don't know if that's too high or not, but yeah, for 3 to 4 sets using 10, just something very traditional because it hasn't been shown to work. And I said that you'd want to use movements that had a large muscle mass, such as like Olympic lifts and things like that, because the more muscle mass that you recruit, the more you're able to recruit that muscle faster so you can increase strength.
Okay.
And then I also said that you'd want to vary your movements from every couple of weeks, and I guess it's called periodization. I did not know that until this this lecture. So that way you can train for different actions because they are all the strength adaptations are specific for a single type of action. And by changing up the activity that you're doing, you're able to better train for all types of movements that you might have to interact with. And I said working both concentric and eccentric movements will help gain even strength gain, although eccentric would be very beneficial, but you destroy your body if you really overworked those.
Okay. Okay.
So that's my answer.
That all seems very reasonable to me. Anybody else have something kind of markedly different or anything really different that you just really want to share?
I said using compound exercises, and I said all the bros in the gym love their isolation, bicep curls, and leg extensions, but compound exercises are so good. One, because you're using a lot of different muscle mass, like squats. You're using more than just your legs. You're using your core too, and even a little bit engaging upper back. But also because the compound exercises are going to be utilizing your stabilizers and your neutralizers, which you might not be using in those isolation exercises. So one, that's just good for body health, but two, you have to use something for that little stabilizer to hypertrophy. So if you're not using an isolation exercise, it's going to be small. So
Megan made many sort of wild, outlandish statements right there that I take any number of I don't know that I take issue with them, but I feel like we need a further explanation. What is a compound lift? What is a stabilizer and a neutralizer muscle? And why would you want to talk about the fact that what if doing the compound lift, what I'm actually doing is I'm lifting only at the capacity that my stabilizers and smaller muscles will allow me to do, and I'm not actually able to tax the primary movers to the same extent that I might if it was an isolation exercise. How would you respond to that?
I would say one, I still included isolation exercises, but make sure you hit the compound. In my little thing I typed up, I focused a lot looking at like squats versus like leg extensions because those are two of the easiest ones to compare. God, I'm trying to remember everything you just said. The stabilizers and neutralizers.
What's a compound exercise?
Oh, something that uses 2 or more joints are moving at one time or during the exercise.
Okay.
Yes.
Why do you think? Is there another reason why that might be beneficial? It has nothing to do with physiology or anything else.
I mean, performance.
Why might it be good for performance?
Because most of the things you're doing in a sport utilize 2 or more joints at one time, like running.
Specificity, right? Specificity. Okay, good. You may continue. Sorry.
I don't remember the other stuff you asked. It was a lot.
It was a lot. It's not me if I'm not asking a 14-part question, right? So, like, what's a stabilizer muscle? What's a destabilizer muscle? I've never heard of those before.
I didn't say destabilizer. I said
What did you say?
Stabilizers and neutralizers.
What the hell's a neutralizer?
I don't remember exactly the difference, but they do different things.
What are we doing here?
Anyone else want to back me up? I'm getting murdered over here. Come on, Keldon. I know you know this stuff too.
What is Jay teaching you guys? I've never heard the word neutralizer.
This is Baylor stuff, so maybe you guys don't learn it. I don't know.
Baylor stuff.
Yeah. But if
so
like in a squat, obviously the prime movers are your quads and your hamstrings and your glutes, but like stabilizers and neutralizers be your abductors and adductors. And then even looking at some of the spinal muscles are helping to stabilize and neutralize your core during it as well.
But why would I want to neutralize something? That doesn't seem right. Also, your
according
hamstrings
to
are
Google,
primary mover during a knee extension?
according
You
to
don't-
Google, a neutralizing muscle provides important support during exercise to prevent injury and restrict movement. These muscles are often smaller than large moving muscles in your body.
So is a neutralizer just a particularly like specially named kind of like synergist or stabilizer muscle?
Probably. That's what it sounds like.
I don't know. Okay, well,
Yeah.
It's because you
And you
did
don't want
anatomy.
leaky energy.
what, Keldon?
And you don't want leaky energy. You don't want any energy leaking out from any joint that's unstable. Like if you're completely, the force has to transmit through everything. And if you have anything that's unstable within any particular joint or muscle, then energy is lost there and you can't transmit it into the ground or whatever and produce force. In my
Wait, so
thinking.
my knee is leaking ATP.
That's a very loose terminology of leaky energy, but like not able to fully transmit the force throughout the muscle into the ground.
I think I know what
That's
it
a
is. Now we're
terrible,
talking
like,
about now we're talking about open chained and closed chained exercises, and I just give up. But I'm like, okay, I just what else? Okay, I understand you're talking about mechanical efficiency and things of that nature, and it's more like mechanical energy, not like my knee is actually. I know what you're talking about. I do all of this because I want everyone to appreciate that precision in your language and precision in what we call things is really, really, really important. And so sometimes people call things stuff that I think is stupid because it's not actually describing what's going on. But okay, so I need to apparently go read up on neutralizing muscles according to Megan. But you never answered my other question, Megan.
I don't remember what it was.
What if I'm going to do some kind of an exercise? Maybe people don't do this anymore, but let's just say that like I'm going to do squats while I stand on Bosu balls or something. Okay. It's called in smiles. I
Okay.
can't lift as much weight while I'm doing that as I could if I was on stable ground. And so my question is, why would I want to do that? Now I'm testing something very different. And so sometimes we got to be careful about, I'm not actually able to overload the primary movers sufficiently because I'm being limited by either the strength or the function in my stabilizer muscles or something else, right?
Yeah, but I don't understand why people want to squat heavy on Bosu balls. It's stupid.
Okay. Well, you can't squat heavy on them. Anyway, there was a time, and maybe they don't do it anymore, but there was a time where I was like, oh, like you need to sit on a balance ball while you do your military press or while you're doing your dumbbell thing or something like this. And, the idea then, well, you're training your accessory muscles. You're not actually training your big primary movers because you're unable to lift. You're limited by these smaller, weaker muscles rather than the bigger, stronger muscles. And you just kind of have to be careful of all of those things. So anyway, it's Alex smiles and Kelvin smirks and everybody. I'm very much just get your ass in the weight room and lift something heavy. Let's go. So, okay. All right, so let's think about question number 2. Is there a genetic limit on the natural, or there is on the natural part of hypertrophy? So we'll start with Pearson. What did you come up with, Pearson? Give me one of the things that you thought might be a natural genetic limit on muscle size.
One thing I mentioned was like the amount of type 1 and type 2 muscle types you have,
Okay.
since that can be kind of genetic. And like if you're squatting, obviously you'll need more slow twitch muscles, and like that could limit, like if you don't have the muscle type to do the lift, then it's going to be hard to do it well and like increase your muscle size.
Okay. That's a very good guess, and it's one of those things that seems very plausible that would be the case. And I can't believe I've taken the slide out, I guess, but what we know is that there actually does not appear to be an appreciable difference in the amount of hypertrophy that we can see in type 1 fibers and in type 2 fibers. And so it's one of those things that it seems like it ought to be the case. And sometimes when you measure people, what you will find is that their type 1s don't get as proportionally as big as their type 2s, but it may also be that they use their 1s a whole lot more, so they've already hypertrophied some, and then we're hitting their 2s kind of at the very end. So that's certainly a very plausible kind of thing, but the actual practice of all of that one doesn't really match up with the plausibility there. But it's a very good thought. Callie, what about you?
Can I add to that? Because I
Please.
think if you have more type 2s, then one of the things that happens is you're able to recruit those higher threshold motor units, which would allow you to be able to contract faster. And I think you can have more force with the type 2.
Yeah, usually you can.
So, I mean, wouldn't that be kind of beneficial for not necessarily hypertrophy, but increases in strength?
So people that have more type 2 fibers, well, but now we're back to talking about, remember that giant list of things that contribute to force production, right? If I have a great big muscle and it's all slow twitch, I'm still probably going to be stronger than somebody that's got a little bitty muscle that's all fast twitch. If we could normalize everything out very, very equally, all other things being equal, then yes, you probably could get a little bit more force out of those kinds of things. You certainly can probably get more force at higher velocities out of them in the midst of all of that. And so your first point is an interesting one, but I would make the counter argument to you that simply having more type 2 fibers wouldn't necessarily make you any better at recruiting that last like 10% of them, right? You at a 50% of MVC contraction may have more type 2s that are getting turned on than a person that's like 90% slow twitch. But everybody, the only way we get good at recruiting those last like 10 or 15% of things is to lift very, very heavy, or to lift a lighter weight very, very near to fatigue, so that we have to learn how to contract those things. And that's a task kind of driven thing. And that seems to be similar regardless of kind of what your normal fiber type is. But those are also kind of good points and good thoughts on all of that. So, okay. Ryan, you got one? Uh-oh. Did Ryan freeze up on us? You're muted, Ryan.
I froze. Sorry. Can you repeat your question?
Would you like to share one of yours with us, please?
Yeah, I didn't know exactly like the route that you were going for this question. So I said like metabolic stress, like
Okay.
how you react to or respond to exercise, like if you're overworking the body or you're stressing it, like how it affects how the muscle grows, and if that like affects like hypertrophy, and if you're doing strength training, like how it affect just like how much pressure you're putting on it, how much stress you're putting on the muscle.
Okay. There's not really a right answer, guys. I'm kind of like what Pearson was doing and that you guys have done. I'm just looking for you to just suggest something and then kind of try to defend why you think that Here's a thing that's probably different among people. Could that be a deal, right? Like, you have different myostatin levels. Okay, pretty simple. Jacob has more satellite cells than Ryan does, so maybe he's got a greater potential. I don't know that any of these things are right, but it makes some level of, like, satellite cells are important. So if you had more satellite cells, maybe that would be good, right? So that's kind of where we are. Okay. Anybody have another one that you just feel really, really compelled that you want to share with us?
I found a pretty interesting paper on, like, or just ran into that randomly when I researched that, about bone structure, which is something we didn't really talk a whole lot about, but it seems like that more bone mass allows you to carry more muscle mass, and then you kind of also have genetic ceiling to bone mineral density, which is improving with resistance training in first place, but there's a ceiling, and that ceiling kind of determines how much muscle mass you can carry.
Okay, sure. I get so big my bones break. That's what I want in life, right? I want to be so ripped up that I'm just like, ugh. Okay. So I'll ask a different question. You guys ready for off the, like, not written down kinds of questions? So these are the fun ones for me. Okay. I won't ask Keldon because he was the one that was probably in my class the most recently. I ask this of my undergrads all the time. So we'll ask Megan because she'll get a kick out of it probably. All right, Megan. Megan, have you ever been to abs class? Or have we done this already?
What's abs class?
Like a workout class where you do 45 minutes of crunches and planks and shit like that. No. Are you aware that these things exist? Doesn't one of you teach like butts and guts or something for us, or body sculpting or whatever in the hell it is?
Tone and sculpt I teach.
Tone and sculpt. Sorry. Oh, such a terrible word. Okay. I think it was at Ole Miss that we had a class that was called Butts and Guts, and it was just like, squat, do crunches, squat, do crunches, right? Do some more crunches and then do some, kick backwards things to work your glutes. So do people in general, if we were to take, maybe not you guys, but if we were going to look in general, okay, would you ever go to a class if they said, all we're going to do is bench press for 45 straight minutes? All we're going to do is we're going to military press. So we're going to do squats or something for 45 straight minutes. No. And yet, I assure you that there will be a bunch of people that would sign up for doing crunches for 45 straight minutes. Okay? And so my question is, why? And is it at all effective?
I think people think that the common misconception is you can spot reduce fat. So they think if they do 45 minutes of abs that they're going to lose fat from their stomach and all of a sudden have abs, when that's really not how it works. But
Okay.
That's what they think.
Right. I got to not go eat at Chick-fil-A so that I can have abs. I agree with you, Megan. But do people, even if they know a little bit better, think about the actual stimulus being applied? So if I go to abs class like that, assuming I can't spot reduce, am I going to get hypertrophy in my abs from that?
Probably not
Okay.
too much, no.
Maybe
It's
not.
probably
Why
more
not?
endurance.
More
Yeah, I would
endurance?
assume that given the resistance you'll be able to have mostly from body weight stuff and the sets and repetitions and doing it for 45 minutes straight, it's going to tax you more muscular endurance than hypertrophy.
Okay. So Keldon, if that's the case, how should I train my abs? What should I do? Also, why would I want to ever do a plank? It's an isometric contraction. We know that isometric contractions, or we do know, I didn't show you guys what they did, but we know that isometric contractions do not result in as much hypertrophy as concentric eccentric contractions. You'd never unrack a weight on the bench and just hold that shit there, right? Never do that. And yet we will plank until we can't do anything. So how might be a better way to train my abs, Kelvin?
Kind of like what we were talking about earlier, maybe some compound movements, and just also through just even basic foundational exercises, you are still kind of training your abs through through your like foundational exercises. You can still get a stimulus through even push-ups, front squats. If you can hold 415 in front of your body without bending over or letting it fall to the floor, then you probably have some pretty good trunk strength.
Sheldon, can you front squat 415?
A little bit more, but that's all right.
A little bit more than that. Jesus Christ. Okay. Okay. Yes, if you can hold 415 in front of your body on a front squat, you've got good core strength. I can promise you that. Okay. Why do people not just do crunches with weight? Why am I not doing a set of 10 while holding 100 pounds in front of me and doing a crunch like that? I'd squat that way. I'd bench press that way. I'd curl that way, right? I'd do a calf raise that way. Why don't I do crunches like that or sit-ups?
I feel like it's because people don't want to get like that much muscle in their stomach because it like gives them the appearance. So a lot of people just do body weight for abs, maybe.
Okay.
I don't know.
I also didn't show you guys some of this data, but the paper that I just showed you guys says that really it's a metabolic endpoint issue. That if I go to failure, if I plank to failure, if I do sit-ups with body weight to failure, I probably have a reasonable chance to get a similar amount of hypertrophy as if I'm lifting a heavier weight also to failure. There's some issues behind all of that, but there's a chance. Right? People are like, oh, I don't want to have these like giant abs. Sure they do. They want giant abs. Like, you know, you see them in there with their dumbbells, like doing their freaking love handle things, whatever those things are called. We won't tell them it's just making it worse, will we, Ryan? So we train our abdominals differently. Can you guys think of a reason, Jacob, why maybe doing isometric contractions to train your abdominal muscles might actually be a good thing to do? Megan's talking with mute on for Jacob.
Oh, well, if Megan wants to talk, she can go ahead. I don't know anything about training. I was just going to make a guess.
That's why we have you guess. That's why we're here. We're learning things.
My only guess would be, well, yeah, you're doing an isometric contraction in your abs. Your ab muscles are a little different than how your other muscles work just because they're layered in 3 different directions. And so maybe doing isometric contraction will work all 3 of the directions that you're wanting to hit rather than going one certain type of sit-up direction or something like that, maybe.
Okay. It's a very reasonable response. Do this for me, Jacob. Stand up. Guess what your ab muscles are doing right now? What kind of contraction do you think your ab muscles are doing while you hold yourself in that posture?
Isometric?
Yep. So guess when Keldon's front squatting 600 pounds or whatever he can do. Guess what his abs are doing? An isometric contraction. Guess what somebody's doing when they're jumping to catch a football or a basketball or something. So a lot of function, actual daily function in the abdominal muscles and your core muscles are isometric contractions. They're meant to be stabilizers and to hold you in some of those things in addition to that. So in that particular way, training them to be better at specifically doing those kinds of isometric contractions is probably a very, very beneficial kind of thing. But again, we don't want to have a gross generalization for our Lululemon girls, right? We all know who they are, right? our sorority girls in their Lululemon outfits that have gone to abs class probably aren't thinking about what they do with their abs all day. They're thinking, I don't know, I'm not going to speak for them. I won't ask anybody to speak for them or anything else, but they're probably not thinking about that, right? Whereas I've been down there and I've seen what Keldon, what they have some of the folks in basketball do and those kinds of things. And there's much more utility when we train athletes and things that we need to do things that are going to be more closely aligned and more closely mimic what the demands of their particular sport are going to place on the body and those kinds of things. So maybe for them, doing planks is actually quite beneficial, or something like all of that. All right. That's all I got. I'll shut up now. Do y'all have questions?
Do you want us to email the assignment?
Yes.
When are
I was just gonna ask when you were gonna post the test grades.
I'm just waiting, Callie, until you pester me enough to post them.
I thought about it, but I held back the temptation.
Okay. I will try to remember. I will try to remember to do that. Okay. So I know I'm sure y'all are anxiously awaiting your test grades and those things, but they're pretty good. So I'm not. Nobody needs to worry.
When are we going over that last PowerPoint?
Next week.
Do we have class on Tuesday?
Yeah. Tuesday and
Okay,
Thursday.
I'm confused because I have finals next week too, and my brain is not keeping things straight.
Because our class is not an evening class, we are on the undergraduate schedule, and so we're going to have class on Tuesday and Thursday just like normal, and then after class on Thursday, I'll give you guys the take-home final, and then it will be due, like Wednesday or Thursday of finals week or something like that. Okay. Of undergraduate finals week. Let me be much clearer in the midst of all of that. Okay. So for next Tuesday, what I want you guys, or what I'm going to need you guys to do is I'm going to post I'm checking now, I'm pulling it up. There should be a couple of papers that I need you guys to look at before we have class. Okay. It's making me log in. Boo. But there's a couple of papers on things that contribute to VO2 max, and then the control of performance and those things, and the thing on critical power that I need you guys to try to read before class on Tuesday. Okay. It will hopefully set the stage for the things that I'm going to talk about, and it will make the discussion that we have on Thursday about things easier. I am pulling up the course right now to see why it's there or not there, if I can get them turned on. And right now, this thing is hating my existence.